So we found that RVD2 plays a very potent role in limiting the progression of atherosclerosis in animal models. And what that means is there are specific histological features of these atherosclerotic plaques in which it limits, such as necrosis and certain cellular populations. We also found that RVD2 promoted remodeling or quote-unquote stability of plaques, which was another feature of protective response and another characteristic of decreasing atherosclerosis progression. I think a lot of the work that we've been doing was based on a strong foundation from the Sirhan group and from several groups worldwide. And what we did was we took a lot of those concepts and said, okay, what are these molecules? How are they made within atherosclerotic plaques? And how are they in relation to other pro-inflammatory lipid mediators? So I think one of the biggest contributions that we made is identifying some biochemical signature of a plaque that is not doing so well, that is highly necrotic and just more advanced in its stage. Since we made that finding, several groups worldwide have looked at this type of SPM to leukotriene ratio as a potential biomarker in plasma or saliva in cardiovascular patients. And I think that was one of the most significant findings we could introduce to the field. So that imbalance is one of that biomarker that I was talking about. And what we found is that in human plaques that are more necrotic or more in their advanced stage, as well as in animal plaques, in murine plaques, that you have this imbalance in these pro-resolving lipid mediators to pro-inflammatory lipid mediators. And what we and many others have done is that it could just be related to the disease and not causative. But what we found is that when you add back resolvins, you can then, during advanced disease, when this ratio is very low, you can actually restore, you can protect the plaque, make it more stable, less necrosis. So I think that's very important because it's not so much just a biomarker, but it's rather a idea or a clue as to how to treat the disease as well. So resolvins that we've studied, we really focus on myeloid cells, which is primarily monocytes, macrophages, neutrophils. And there's a lot of research about a lot of other cell types in atherosclerosis. Our work primarily focuses on those cells. And what we found is that resolvins activate key cells like macrophages to stimulate the clearance of dead cells, for example. There's a large accumulation of dead cells in plaques, in atherosclerotic plaques. That's why you have these large necrotic cores. And if you don't clear these dead cells efficiently, that's a problem. That worsens the disease. So one of the major bioactions that we found is that resolvins can act on these macrophages to enhance the clearance of these dead cells to help remodel the plaque. I think especially in the cardiovascular arena, there should be a lot more emphasis on lifestyle changes. And I'm sure in the clinical practice there is. But there is likely not a magic bullet for any disease, and especially in cardiovascular where you can really modify your lifestyle with diet, exercise, especially early on in life. That I think is extremely important. And trying to understand a little bit more about immunonutrition and what is the best pro-resolving diet, is an active area of research that is not my own, but many others. That would be the best ideal scenario. I do think that SPMs are very clinically relevant. They have a lot of clinical applicability. How they would be treated, I know there was a study from Tom Van Dyke's group a few years ago, or several years ago at this point, where he put SPMs in mouthwash and looked at atherosclerotic plaques. And if you think about atherosclerosis, and he found that the top, this is called oral topical, right on the gums, and he found that atherosclerosis was actually halted in its progression. And if you think about atherosclerosis, this disease it manifests later in life, but it starts earlier in life. And it can start even, just forming fatty streaks along the artery can start in adolescence or even younger. And we all brush our teeth. Instead of taking a pill for the rest of your life, maybe you could take some mouthwash. And I think that would be a wonderful application, especially in a disease that is so progressive. And that's how I could envision it to help a lot of people. Well, our lab, because cardiovascular disease and atherosclerosis is a disease of aging, my lab is very excited in processes associated with potentially dysregulated or failed resolution programs during aging. So we're very interested in understanding how resolvins might help be organ protective in aging, or what mechanisms may derail SPMs or other resolution factors during the aging process. So I would explain it in a sense that in our body, we have cells that communicate with each other. And the way they communicate with each other is by certain molecules, certain factors. And when you have some sort of inflammation or injury, cells communicate with each other and they say something's wrong, something's not wrong. And these SPMs are a way for cells to communicate with each other to say, we need to repair something. Okay? They are made, they're the cell's language to say, hey, we sense something's not right, and let's start this repair. And I think that would probably be the best way to describe that. And the way cells communicate is not only factors, but their ears are receptors, so they are released by cells and they're received by other cells via very specific ears or receptors. And that's how these cells can communicate about how to sense that something's wrong, how to help repair. So SPMs are they're made in your body from essential fatty acids called EPA and DHA, which are primarily in fish. And essential means that you have to take it in from diet. Our bodies cannot make these compounds on their own without the proper diet. And what happens in your body is that when you take these essential fatty acids, our cells have a process in place to kind of break them down. They digest it as we would just digest food. Our cells can also digest these types of essential fatty acids into these SPMs that are very bioactive. That means that they have a lot of strong bioactivity on cells. And the way they act after our cells digest these essential fatty acids to make these potent ligands, these molecules act as a language to communicate with other cells via certain receptors to stimulate an entire program that is distinct to a particular context. Our context is cardiovascular disease, so what these ligands do is activate repair programs in myeloid cells as well as other types of cells. Whether certain individuals can produce or digest these essential fatty acids to produce these SPMs is an ongoing area of investigation. And really, that seems to be where a lot of us are trying to understand. Is this a defect in the ability of our bodies to make these compounds during certain disease states or not? Is it an overability to further digest it into inactive? Are they being actively metabolized? Are they completely fine in their synthesis? Or is it a problem with the receptors to which they bind and and how they signal in cells? And this is an ongoing area of exploration in the cardiovascular arena as well as several other disease states. So whether elderly individuals can produce SPMs better than others or not, or whether cardiovascular patients can, I think we need to do a lot more to get a deeper dive into that. I do think taking exogenous SPMs would be ideal for those who can't produce SPMs on their own. We have not ruled out, there's no such thing as the perfect drug, and there's no such thing as a one pill fits all, especially in humans. And I do think there's a lot we need to learn about responders versus non-responders and who can be most optimal to SPM therapy.